So maybe if you want to. Okay. So the talk is called Plant Diversification in a Desert Archipelago, a Phylogenomic, Systematic, and Historical Biogeographic Analysis of the Rock Daisies, Perityleae Compositae. So this is my thesis while I was working in the Department of Integrative Biology in the Jepson Herbarium at UC Berkeley over the last 5 years. And the thesis was filed this summer. And today the talk is mainly just a celebration and to share kind of some of the findings inside of that thesis. Let's see. So here's our rock daisy roadmap for the talk. I'm going to give an introduction to the system and some of the main questions. I'll talk about resolving the evolutionary relationships in this group using phylogenomic, morphological, and chromosomal data. And then we'll revisit some of the big questions in light of that data. And finally, I'll talk about a new classification of rock daisies that's going to be based on these phylogenomic insights. So an introduction to the system and questions. The desert is the biome that is defined by a lack of precipitation, right? So it's dry and lack of water also means lack of erosion. So the desert is rocky. There's these jagged rocky mountains separated by low basins. So here's what I want to do to start. I just want you to imagine yourself in the desert and you can close your eyes and you can imagine the wind starts to pick up and the wind interacts with the mountains, form clouds. You hear thunder and rain, and the birds start to sing, and you hear spadefoot toads, and you open your eyes to a whole different place. So as a biologist, this is one of the most amazing experiences you can have. One of the first things that you learn in a biology class is that life is wet. We are made of water, and we need water to survive. So I can't think of a better, more remarkable obstacle that life has overcome in its 3 billion year history than being able to live in these extremely dry habitats. But deserts actually, they contain this really diverse and really interesting, unique flora that's adapted to live in dry places in one of two ways. So you have those plants that escape drought. These include the ephemeral wildflowers that hide in the soil and they sprout and they grow, they go to flower and seed really quickly during periods of good weather. And then there's those that tolerate drought. And these are the plants that have interesting growth forms, or they have deciduous habit. They have succulents or other strategies to live through the very dry periods. And if these are some of the iconic examples of evolutionary adaptation. But when you start to look into it, it's actually, the question of where these adaptations came from is something that's puzzled evolutionary biologists for a very long time. And it goes back to Ledyard Stebbins, the experimentalist and one of the architects of the modern synthesis, who wrote this paper, this very influential paper, Aridity as a Stimulus to Plant Evolution, in which he argued, among other things, that the interesting growth forms and the diversity found in dry regions came as a result of a response to aridity. So I call this the adaptation hypothesis, and we can sort of draw from it a set of simple predictions. If you imagine a clade of desert plants here, and at some point there's a shift into a dry habitat, or the habitat becomes drier, then in response some adaptation arises that then allows the plant to diversify in that new habitat. On the other hand, an alternative hypothesis that was described by Daniel Axelrod that I find really interesting and called the pre-adaptation or ex-adaptation hypothesis was influenced by his work in paleobotany. So he worked on fossil floras all over the Southwest. And one of his big discoveries was that although arid biomes like the desert, they cover a huge extent now, they're actually very young on a geological timescale. So the Earth has been getting cooler and wetter over the last 10 million years. And deserts as we know them today didn't really exist that long ago. In fact, more than 2 million years ago, they would have been found in pockets here and there. And more than 5 million years ago, it would have been a little bit difficult to find deserts. Going back any farther, dry habitats would have been restricted to places like rain shadows of mountains and valleys and edaphically dry rock outcrops. So Axelrod reasoned that given that the desert plants, they seem like they must have come from great antiquity, that they may have evolved in these little microclimates. And then when the environment became more arid, they spread and diversified, kind of taking advantage of that new opportunity. So we can illustrate this similarly. You imagine a group of focal desert clade, but some arid adaptation is already present. And then when arid conditions occur, or when the group disperses into an arid location, then diversification follows. So these are two kind of alternative hypotheses with clear predictions, and could be easy to test. But it's actually a little bit harder than it seems, because what you need to do here, you need to have a group of desert organisms that you can comprehensively study. You can understand their relationships. And then you also need to trace the history of how they dispersed into or shifted into dry areas, into deserts. And you need to understand and characterize their traits and the traits' relationship to their ecology. So this has been a motivation for me, and why during the last 5 years, I've basically tried to learn everything I could about this one really interesting group of composites called the rock daisies, the tribe Perityleae. It actually turns out to be a really good group for asking these kind of questions, because it's a clear monophyletic lineage with that is diverse, but not too diverse for a tribe of Compositae. There's 7 genera, and they're all united by some clear morphological similarities, including this 4-lobed disc corolla and 1 or 2 series of subequal boat-shaped or naviculate phyllaries that are often glandular, and cypselae, fruits that have 2 or 4 sides, usually callus or ciliate margins, a crown of scales, and some bristles. And they were really carefully monographed in the late 60s and early 70s by Michael Powell, which gives kind of a really great foundation. Another reason why they're really interesting for this kind of questions is that they include some of the most abundant wildflowers in those ephemeral blooms that take over the desert in good years. But they also include some shrubs that are found in more montane settings, and then some that are found on isolated islands. But this group hasn't really been the focus of a molecular phylogenetic study, at least a densely sampled molecular phylogenetic study, even though it seems really good. So you wonder why? Well, it turns out that a majority of the diversity in the group are these suffrutescent perennials that are ecological specialists on extremely sketchy and dangerous, difficult to access vertical rock cliffs. So that's kind of repelled a lot of people. They actually, it's pretty amazing. They have these woody codices, these perennate organs that actually root into minute crevices in the rock, and they're able to grow there and resprout from that perennial root base from year to year. So luckily, for the first 2 years that I was studying these plants, I really didn't even have to climb the cliffs. I used herbaria, and herbaria, where the collections of past botanists are housed, are just this amazing treasure trove, this amazing resource for studies on plants. And I'm really grateful to all of the herbaria and the personnel who worked there that I was able to visit. I visited big herbaria and I visited small herbaria. They were all very useful. And I had help from many people along the way. So I want to acknowledge and thank those people. I also did a lot of field work. So kind of at the base of this project was 13 months, more or less, of full-time field work. And essentially what I did was roll across the range of these plants in a pickup truck and try to climb up the different desert mountains and encounter as many of the plants as possible to fill gaps in sampling. When I found plants, I made herbarium specimens and I collected leaf tissue for DNA analysis. But I was rarely alone. I was helped, on this journey by many, many, many people, more than who are shown here, including 6 undergraduate Berkeley students, my research apprentices, who were able to join me in the field on this project and get some training, and many really, really talented botanists on both sides of the US-Mexico border who were welcoming to me and were willing to go to the field with me. So thank you. Before I deep into some of the results here, I also want to acknowledge the complicated history of both extractive and settler colonialism that is fundamental to the range of the rock daisies, and that has affected multiple indigenous nations, including those listed here. I want to acknowledge these first peoples and also the Wichin Ohlone, the original stewards of the land we call UC Berkeley. But beyond acknowledging kind of this past, I also want to give this link here to Sogorea Land Trust, which is a place where you can go and you can actually calculate a land tax that you can used to support rematriation of indigenous land. Okay, so part one is resolving relationships using phylogenomic, morphological, and chromosomal data. So here, I'll just kind of give a broad sampling of what there is here, including our phylogenomic approach, reconstructions of chromosome number change in the tribe, and morphological evolution. A lot more details than I'll cover are included in this paper, which we published in a special issue of the Journal of Systematics and Evolution, dedicated to Vicky Funk, the late curator of Compositae at the Smithsonian Institution. And this was, something I'm really proud of, because Vicky was my mentor and my advisor as a pre-doctoral fellow at Smithsonian. And she actually developed, with Jen Mandel and others, the approach that we took in this group. We applied what they called the Compositae conserved ortholog set, which is a hybrid enrichment approach for capturing 1,000, more or less 1,060 low copy nuclear loci from across the genome. And the way you do this is you use baits specifically designed for Compositae based on some available genomes. And you get more than 1 million base pairs of phylogenetically informative and putatively orthologous loci. Obviously, there's more details in the publication, but here I'll basically explain that we compared the results based on these COS loci to results based on chloroplast DNA and ribosomal DNA, chromosomal patterns, and morphological data. We really found that the COS loci were really effective for resolving relationships with high support, for finding monophyletic groups within the tribe, and that in general, this data was really nicely congruent with nuclear ribosomal DNA and chromosomal data. That chloroplast data was a bit more complicated, but I'll show that when we reconstructed chromosome evolution using chrome-eval, one of the things we found is, a typical kind of base chromosome number for the Compositae of X equals 9. That in this group, you have a base chromosome number of X equals 18 that's retained in some of the earlier diverging lineages. And then nested within those lineages is this interesting group that has a lot of chromosomal variation that is the result of a lot of disploidy, so gains and losses of chromosomes. But that the majority of taxa in the tribe, they all fall out in this one group that has a stabilized chromosome number of X equals 17. And that's the result of a single disploidy event. A lot of neopolyploids are found in this group, so kind of terminal taxa that have duplications of chromosome number. And the X equals 17 group, this is the last thing I'll show here. The X equals 17 group and the group with a lot of disploidy actually form two subclades of the core genus Perityle, which was found to be non-monophyletic, with each of these clades kind of forming separate geographic, morphological, and chromosomal clusters. So this is a reconstruction of morphology. So in here, we focused on those morphological traits that were emphasized in past taxonomy, and we reconstructed their evolution onto the phylogeny, and that's what's shown here on the left. We also used a reversible jump MCMC approach to kind of figure out the different transition rates between the morphological states. So here you have reconstruction of white ligules, white ray florets, and yellow ray florets across the phylogeny. And you can kind of see that the different colors come out in accordance with major groups with some, trickiness there. And that's reflected in this graph on the right, which shows the transition rates, which are both positive from white to yellow and back again, but very low. So flower color turned out to be one of those traits that was pretty conserved across the phylogeny. Here's a different example. So fruit traits, the presence or absence of different pappus elements and shape has been one of the things that was emphasized the most in past taxonomies. But we found that fruit traits in general were more dynamic than previously thought. So here, reconstructing ciliate margins along the outsides of the fruit, what we find is that they were ancestrally absent in the group, but have been gained many different times. And that's reflected in this graph here. The green representing gains has a positive transition rate, and it's not readily lost after it is gained. That low amount in blue is shown there. One of the most important taxonomic characters has been the presence or absence of these pappus bristles. And what we find here is that pappus bristles have been lost multiple times throughout the group, throughout the evolution of this phylogeny, of this group. And in one case, they've actually experienced kind of an acceleration from few to many bristles. So kind of looking across many morphological traits to sort of summarize, what we see is that fruit characters were more homoplastic than previously believed, that flower characters were relatively more conserved, but there were multiple independent losses and gains of a lot of these traits, in fact, in all cases. So I think this is interesting, but what's going on here? Well, you have to remember that these plants live in very extreme environments. They live isolated on these rock cliffs on tops of mountains. And so there's a very intense ecological pressure on these plants for dispersal. To get from one isolated rock face to another, the presence or absence of a pappus briar might make a big difference there. But it also goes the other way. Because they're isolated on these habitats, if they fall off the cliff, they die. So you can imagine there's really strong selection for gains and losses of fruit traits. And that obviously that can be confounded by other things. They live in very arid habitats, which can have a big effect on fruit size. But there's certain behaviors that really convince me that there's kind of an island-like loss of dispersal going on here. One of those is that when some of these plants that live on cliffs, they have this thing they do where as the capitulum develops, it actually bends back and it plants the seeds back into the cliff face. Okay, so main takeaways here: target capture resolves uncertainty in the Perityleae radiation. And disploidy as well as neopolyploidy are revealed as important processes of chromosome number evolution. And morphological traits are found to be more dynamic than previously believed, which implicates, ecological pressures. So now with this new data, we can revisit some of those big hypotheses, including, distinguishing between the adaptation and pre-adaptation ideas. And really what this comes down to is figuring out how and when did the rock daisies shift into their desert habitats, and how and when did the ecological specializations that they have for desert habitats come about? So I'll mention that we, now knowing that the target capture approach worked really well, we went back and added 74 more samples to get a much more comprehensive idea. And the final sample includes 114 accessions covering more than 95% of the diversity of recognized taxa in this group. And there's multiple samples included here for any of those that are geographically widespread or that show morphological variability. So we're not relying too much on the taxonomy. We found further support for relationships that we found in the previous analyses, including most notably the non-monophyly of this core genus Perityle. So we produced a time-calibrated phylogeny to get a handle on the timing of the events, to get at these big questions. And getting a dated phylogeny is fundamental to a lot of these other questions. But estimating divergence times is kind of an art, and I think it's important to evaluate it with multiple criteria. But I'll say that there are some things about the dates that we got from these kind of analyses that convince me that they're pretty solid. One of those is this split early in the phylogenetic tree of Perityleae, with one of the clades including many members that are found in the Baja California Peninsula, and the other clade including a lot that are centered on mainland northern Mexico and the southwest US. And it's interesting that in timing, this split corresponds with more or less when a proto-peninsula was slothed off of central Mexico, began to float north to eventually form the Baja California Peninsula. And if you actually model biogeography to this phylogeny using a DEC model, you find a cladogenetic split here corresponding to kind of that early part of the phylogeny. I'll also point out this group here, which I'll call the Laphamia lineage. And this one is really interesting because if you begin to look at the onset of aridification in arid habitats across the timing of this tree, you see that most of the major divergence events happen prior to aridification, except in this Laphamia lineage. And that is the group that contains a lot of the desert plants. Okay, so with the dated phylogeny, the next thing, and this is kind of an important one, so I'll walk through this. what I did was reconstruct the shifts in biome across the group. And I did this using a biome shift model that takes into account 4 different major habitats: desert, tropical or tropical deciduous forest, subtropical forest, and temperate forest, as well as 4 areas: the basin and range. Here's a map. Basin and range, the Sierra Madre Occidental of northern Mexico and the transvolcanic belt of Mexico, Baja California, and South America, as well as all the different combinations of those biomes and areas. This was also an analysis that included time slicing to kind of model the growth of more arid conditions across the evolution of the group. And what I want to point out, there's lots of interesting stuff here, but what I mainly want to point out are these shifts into the desert biome. First of all, there's many independent shifts into the deserts across this group. But one of these shifts accounts for more than 85% of the contemporary diversity found in the desert, and that's within the Laphamia lineage. The other shifts happen mostly in terminal taxa, and they are all resolved as more recent. And this is interesting because, let me also say one other thing about this, which is that the shift in Laphamia into the desert happens out of these tropical deciduous forests in Northwest Mexico, and there's a shift into the basin and range, kind of larger North American deserts there. And this is where the kind of particular natural history of the group comes into play here. So we want to understand where their ecological specializations within the desert come from. And I mentioned that they often live on bare rock environments. And when they do, they tend to have a particular life history. They have this kind of perennial codex. And so what's interesting is that if you find a strong association between microhabitats and traits, then we can start to understand kind of the historical microecology of the group. And we do find a really strong association between life history and microhabitat preferences for adaptive specialization. So looking and actually reconstructing those ancestral ecological specializations onto the tree, what we find is that the group has an early switch to the bare rock endemism that was readily lost multiple times, but very rarely regained. And I'll point out that that shift into the desert biome that occurs in the Laphamia lineage happens from a group of bare rock endemics that is found within a more densely vegetated kind of tropical context. So obviously we can't go back and see what those ancestors looked like. That would be great, but what we can do is we can look at contemporary lineages that live in this kind of habitat, which looks like this. So you can kind of compare it to your image of the desert that you got in the beginning here. This is tropical deciduous forest, and it has reliable precipitation for part of the year, which, precipitation means erosion. So there's a lot of decomposed substrates, and they tend to be covered in a really dense kind of, distinctive forest. But in places, there's outcrops and openings. And in those outcrops and openings is where you find these really interesting Laphamias. So what it appears happened here is that you have this lineage of edaphic specialists that was living, kind of in these unique habitats with growing aridification. That's the lineage that spreads and has this amazing radiation throughout the basin and range onto a lot of heterogeneous habitats, rocky habitats, and drier habitats. So the major takeaways here are extensive diversification in the desert rock daisies originated from ancestors that were pre-adapted for dry conditions as edaphic specialists on bare rock outcrops and more densely vegetated biomes, and that a shift into the basin and range deserts about 5 million years ago led to a recent radiation in the sky islands of the arid Southwest US and northern Mexico. And so I think this is really cool for several reasons. One is that I think it's a really good empirical example, kind of supporting Axelrod's hypothesis that desert lineages gained their unique specializations and life forms in microclimates and then sort of took advantage of deserts as they began to spread and appear. But I also think it's kind of an interesting thing to look at in many other groups of plants, because when you look on these cliffs, you don't just find rock daisies, you also find relatives of other diverse lineages of desert plants. like these chilanthoid ferns and these cacti shown here, but there's many more succulents, legumes, agaves that live in these habitats. And in a way, it kind of makes sense. If you look in the literature, it's sort of hard to find examples where aridity, climatic aridity leads to huge changes in plant form or plant growth. But there's tons of examples where edaphic environments lead to catastrophic change in plant morphology. This slide is for you, Bruce. There's also another element here, which is that, you know, some people will probably find evidence for and will claim that dry environments were present earlier than you know, Axelrod claimed. And I don't doubt that. But we also know that the climate has shifted a lot and that dry environments likely arose and then disappeared and then arose again. It's hard to imagine that they were constant over time. But it is very easy to imagine that rock cliffs, which have been present and constant since the Eocene, may have been an important, you know, place for arid lineages to go, kind of like a shelter during wetter times. Okay, so the last part here, I may need to check where we're at on time. If somebody has the time, can't really see it. Well, I'll just go. Somebody let me know if I'm going over time. Oh, perfect. Thanks, Jason. Then we're good. We can talk about this last part here, which is a new generic classification for Perityleae. And what I want to just, you know, what I want to do here is give an overview of the new treatment. This is the first treatment, taxonomic treatment of Perityleae to be based on a molecular phylogenetic, densely sampled kind of phylogenetic framework. And I want to give a panorama of some of the novel groups here, including a new narrowly circumscribed Perityleae, Nesothamnus, Galenstogiopsis, and Laphamia. So the goal of this here is to more or less resolve the non-monophyly of that core genus Perityleae. Luckily, there's an abundance of names already available. So what we did was kind of make new combinations in order to present a generic taxonomy that aligns with monophyletic groups. And present a dichotomous key to genera and a general natural historical overview of the various groups in light of all of this new data. So one part here is a new narrower circumscription of Perityleae to include an early diverging lineage with 13 taxa. I mentioned this a couple times throughout the talk. What I'll say here is that there's, in this more narrow circumscription, there's less taxa in Perityleae, but it still remains the most heterogeneous morphologically in terms of chromosome numbers and geographic range of the genera in this tribe. The type species of Perityleae is this one here, Perityleae californica. And it's part of it forms a group with Perityleae cuneata, which has 2 recognized varieties, but they kind of fall out in an interesting pattern. And there's some interesting natural history here where they have what you would call a descending disploid series radiating out of the Sierra La Laguna, the sky island at the southern portion of the Baja California Peninsula. And that descending disploidy corresponds with environments with more arid conditions. The other group here within Perityleae includes the members that have white ray florets. And included also in here are the 3 taxa of the genus Amaria, which have a lot of morphological similarities and come out as a group with these white rayed Perityleae, including Perityleae crassifolia with 2 varieties and Perityleae emorii, as well as others. And this group is also really interesting because, as I mentioned, the range of Perityleae includes these long distance amphitropical disjuncts in Chile, both in the Atacama and on the isolated archipelago, the Desventuradas archipelago. And these taxa actually turn out to be nested within Perityleae and are recognized as Perityleae tenuifolia. And interestingly, the plants in the Atacama have been recognized in the past as Perityleae emorii, a widespread North American lineage, but they don't form a monophyletic group with us, so they likely need to be recognized as a distinct lineage. And also in this group is Perityleae socorensis, which is endemic to the Revillagigedo archipelago off the coast of Colima, Mexico, which is south of Baja California. We also reinstate Nesothamnus in canis, a really interesting plant endemic to Guadalupe Island, an oceanic island off the coast of Baja California. And this one is really interesting. It comes out on a long evolutionary branch, and its chromosome number suggests, along with its really interesting and out there morphology for the tribe, that it may have allopolyploid origins. It's one of the only plants in the group to have these dense capitulescences of many heads, and it has this really distinct kind of canescent and dissected leaves. So there's some evidence for an interesting allopolyploid history, but also that these unique traits may have evolved, consequent to dispersal to an isolated island. So this is one that I'm really excited to, learn more about. Reinstatement and expansion of Galansogiopsis as a clade with 17 taxa is another part of this. And these are plants that have white rays and fruits with ciliate margins, as well as a crown of scales, that are centered in the Sierra Madre Occidental mountain range in northwest Mexico. Included in this group is one annual, Galansogiopsis spilanthoides, which is formerly called Perityleae microglossa. And this is really the one self-compatible annual in this group that is very common, has a very wide geographic range, and it grows in a variety of habitats, including archaeological sites and decomposed substrates. And here it is growing in the wall of the cathedral in Tequila in Jalisco, where Sophie and I were doing some fieldwork. There's nested in this group this really interesting clade that seems to have undergone a really rapid radiation onto isolated rock cliffs within a subtropical forest context in the southern Sierra Madre Occidental that has a lot of really interesting and possibly undescribed diversity. And there's also members of this group that make it into Arizona and New Mexico. Galansogiopsis ciliata and Galansogiopsis coronapopholia are two of those. Here is Galansogiopsis coronapopholia growing out of an altar in Patagonia, Arizona. And these grow on volcanic substrates mostly, sometimes limestone, and are sort of scattered around. And you can see Galansogiopsis turns out to be a pretty apt name for these because they tend to look superficially like Galansoga, a pretty common composite weed. A big part here is reinstatement and expansion of Laphamia, the Sky Island rock daisies, as a clade with 57 taxa that have undergone an extensive radiation throughout the basin and range. And these have yellow rays present and usually have fruits with reduced pappus. And what's really interesting about these is that they kind of form lineages in different archipelagos within the desert. So I mentioned a little bit these that grow in tropical deciduous forest in northwest Mexico. And these are really interesting plants, and I've really enjoyed studying these. They're very beautiful, kind of classic daisies. In the Chihuahan Sky Islands, there's a really interesting group that has a lot of morphological change, including different flower colors and leaf shapes, as well as some interesting pappus element kind of things going on, where some of the fruits have developed many, many bristles. In the Great Basin, there's another very diverse and very interesting group that tends to have a lot of ecological kind of differences going on. There's plants found on limestone and others on sandstone and volcanics. And this is an area where there are a lot of isolated mountain ranges, where there's been a lack of botanical collections, and where there's a lot of really interesting things yet to be studied. I'll also point out that in the process of making these combinations, there were some places where we wanted to make a combination, for example, for Perityleae palmeri, but that was already taken within Laphamia. So we had to create a new name. And so the one that we're recognizing is Laphamia sanchezii, and I'm really proud to make this name for Jesus Sanchez Escalante, the curator at the Houston Herbarium and organizer of the Red de los Herbarios del Norte, which is a really amazing consortium of herbaria in northern Mexico that is doing a lot of really amazing stuff. And I also want to mention Laphamia inuensis. So Laphamia as a group, as a recognized group, may be one of the genera in the Southwest with the highest proportion of rare or threatened taxa, according to NatureServe. One of those is a California endemic to the southern Inyo Mountains, Laphamia inuensis, which is threatened by gold mining on conglomerate mesas. Just a project that has been pushed through during the Trump years and then has kind of just been continuing to gain steam. And there's a lot of resistance to this project. And so I would encourage everybody to go to protectconglomeratemesa.com. You can say you heard this talk and you got excited about the rock daisies and you don't want this mining project to stamp out a California endemic rock daisy. Okay, so this is just been kind of a broad overview of a massive project, and I wish I could just go on forever telling lots of stories and talking about a lot of things, but really this project only began to scratch the surface of this group. You can tell, we're working at the broadest level here. So I'm really excited to continue to generate phylogenetic information at finer scales and start to, ask some, to explore some of the really interesting questions in this group and keep an eye out for new species descriptions. One of the things I'm most excited for is to get in there and begin to dissect the mode and the tempo and the mechanisms of diversification within the Sky Island lineage, within Laphamia, which is going to be, a longer term kind of endeavor, but really interesting. So with that, I want to thank everybody for listening. I hope that I didn't, push it too long on time, because I want to give everyone a chance to ask questions or, just to share stories or anything like that. But I have to thank my mentors, especially Dr. Bruce Baldwin, who was a perfect advisor for me during this project and who gave me tons of latitude for independence, but was there when I needed him, as well as my other mentors and my students who helped with the amazing, incredible amount of and field went into this project. I want to thank my family for supporting me. And I want to thank those who helped me in the lab, especially Gabriel Johnson and Lydia Davis were really, really helpful. With bioinformatics, Mike May, Carrie Tribble, and Michael Landis. I want to thank you. And then I also want to thank Will Freeman and Adam Schneider, who kind of began this project with me. They were my lab mates in the Baldwin Lab. I want to thank all the organizations that provided funding for this project, which was kind of scrapped together with small grants from many, many, many different sources. I want to thank the Smithsonian Institution and the Department of Integrative Biology at UC Berkeley and the UC Botanical Garden for really supporting me throughout this project, and the Philomathia Foundation as well. And then I also want to thank everybody who, just basically pitched into this project and gave me information about where to find plants and where to go and where not to go, and who, was just helpful in a million different ways. So I think that's all the gushing that I'm going to do. But Bruce, if you want to add anything, or I can continue sharing my screen and leave this up here and kind of open up to questions now.